Hello and welcome to the Deep Sea podcast, Pressurised, a short, punchy version of our main feed that gets right to the scientific point. If you like what you hear, you'd like to hear the full episode, you can find it in the same feed. And now, to get right to the point. There was some nice footage from Mbari at about 1,400 metres down, so 4,500 feet off the California coast. They saw a squid brooding her eggs.
Ooh.
And it's unusual for squid to brood eggs. They tend to either release them as floating mats or to plant them somewhere. But there are some examples, and interestingly, all the examples of squid brooding their eggs are deep sea. So it's obviously a viable strategy when you're reproducing in the deep sea. So the species was Bathyteuthis berrii, and there's some nice papers on it. So I'll put those in the show notes. Do you have any more newsy stories? Or...
The only other one I saw which was interesting was discovering why deep sea corals glow in the dark. Well, yeah, it's a bunch of scientists from Tel Aviv University are saying it's a lot of these corals give off fluorescence. So they use the downwelling light to excite pigments and give off these beautiful displays. And no one was really quite sure what they were for, but they reckon it's to attract in plankton, signalling their prey.
And they did some quite cool lab experiments, didn't they? There's like colour and pattern preferences to little prey invertebrates. Really weird. I'm quite enjoying the sort of paleo stuff. I'm quite enjoying the looking back in Earth's history. So the deep Atlantic was once as warm as the Mediterranean. It was 20 degrees 50 million years ago. And so ancient temperatures were reconstructed from the stable isotope ratios of critter shells in the deep sea muds. So coccolithophores, things like that. You get a nice 3-meter sample of deep sea mud, and it's essentially going back in time, basically. So that's going to help us understand elevated CO2 and the impact on our planet. But 20 degrees, in all of the Atlantic.
That's mad, isn't it?
So there was a new brine pool discovered in the Red Sea, Gulf of Aqaba, the small inlet to the north of the Red Sea. The main pool is huge. I thought this was a typo at first, but 10 square kilometres, and then with 3 minor pools around it. Well, they're quite excited because they preserve the sediment really well, because of course they're pretty abiotic. So they reckon there's 1,200 years of geological history beautifully preserved at the bottom of it in its sediments.
Yeah.
Very, very cool. Did you hear about the Java Sea off Indonesia glowing back in 2019?
Glowing?
Milky Sea. This one was Java off Indonesia. It was more than 100,000 kilometres squared of the ocean was just glowing.
Nice.
And the satellite observations were published last year. And following the publication, the crew of the yacht Ganesha got in touch to say they were there. And they had photos from the surface, and they sort of did a little bit of citizen science and tried to sort of figure it out. So it was a really nice example of ground truthing of satellite data, really wondrous things still being discovered.
Anyway, so all this talk of fluorescence and bioluminescence and going from a whole episode on mesopelagic fish and other bioluminescent oddities, we should conclude this, don't you think, with a trilogy, you might say. We need to follow up with someone who knows about the eyes themselves. So we should phone someone up who knows about that.
And who are we going to chat to?
I want to chat to Justin Marshall. Right, so in the last few episodes, we've learnt quite a lot about things like the mesopelagic or the deep pelagic, and then we were talking about bioluminescence. But the final piece of the puzzle is vision in the deep sea. Today, we have Professor Justin Marshall, who I was going to say from University of Queensland, but I think you're now retired. So, Justin, how are you doing? Welcome to the podcast.
Oh, yeah, good, Alan, thanks. And I'm sort of retired, but believe it or not, I'm at a conference. You can still say I'm at the University of Queensland. And yeah, still happily studying fish and wondering why they have eyes.
Excellent. So what is unusual about deep-sea fish eyes, the adaptations and so on?
The answer to that is actually not much. They're pretty much like fish eyes from the surface. They're pretty much like our eyes. Now, our eyes are called simple eyes because they have, the cornea, curvy bit you see at the front of our eyes, and that focuses the light onto the lens that focuses the light onto the retina and makes our image nice and sharp. In the sea, the cornea doesn't do anything because the cornea is sort of the same refractive index as water. So fish's lenses are a bit different. But, whether it's a fish on the surface or a fish in the deep sea, they have these spherical round lenses and they have a retina at the back. So really the answer to the question is not that much until you start thinking about it. And then there are things in the deep sea that you need to do that you don't need to do on the surface. To cut a long story short, there's quite a few eyes that have become more simple. And then there's weird eyes that sort of have divided into two, and other eyes that have become more complicated. So they go in both directions.
So the four-eyed fish, for example, is that something which is very specific deep sea, or is that evolved in any other animal?
No, that's very deep sea-ish, and there's a whole bunch of those. And the reason for that is that if you think about these fish in the mesopelagic, and of course, as we all know, as soon as you get below 1,000 meters in the ocean, there's no light left from the surface. So it's all bioluminescence. But when you're in that mesopelagic realm, let's say at 200 meters, there is light from the surface. And then as you sort of think about looking out to the side and looking down, it gets darker and darker. But there's still stuff there. There's still the bioluminescence. And lots of the deep sea fish at that level will have eyes that point upwards. So there's a famous fish called Apisthoproctus. Its main eye looks upwards, but then it has this, or its relatives have this series of adaptations that then allow the fish to look downwards too. It's almost as if it sort of forgot about that bit. So it designed most of its eye to look upwards, and then it went, oh, hang on, I do need to see stuff down too, because that might be a predator coming towards me. They sometimes add what looks like a complete eye that's looking downwards. It's called a diverticulum. Some of them have their own little lens and their own little retina. And others have these sort of lens pad things that reflect or refract light into the eye, allowing the fish to have this sort of vision downwards as well, while focusing on the light from above. So it kind of covers all angles, if you like.
So the thing that I was interested in, the vertical component to that, of course, the sort of downwelling light is one thing, but let's say you're completely beyond that. You're at 5,000 meters on the middle of an abyssal plain.
Yeah.
Something flashes. How far away are things seeing that? I mean, because we keep talking about bioluminescence is used for communication, and then everyone runs off a list of potential things it could be communicating, but how far is it emitting that, do you think?
Oh, it's a good question. One of the things that a lot of people don't realize is that vision in the ocean is limited by water. So no animal in the ocean is going to see like we do, 100, 200, 300 meters away, a kilometer away. We can still spot things miles away because air doesn't attenuate light very much, or not as much as water. Whereas water, even if it's very pure, will cut light down. And then as soon as you start adding stuff to it, like particles or sediment or dissolved organic matter, then it attenuates very quickly. So most action in the ocean, whether you're in the deep sea or in the surface, is over just a few meters.
Is it as short as that, really?
Yeah, I mean, it's a surprise. A lot of animals that live in the surface waters, especially, as I said, when you've got this organic matter in there, their whole life is conducted within possibly the space of the room which you're sitting in. In the deep sea, the water does tend to be quite clear. So it can be 100 meters, that sort of thing. But that's a long way away for a little fish. It's quite difficult to judge distance in the sea also, especially in the deep. You see a flash. Now, was that a big flash a long way away? Or was that a little flash very close? Imagine sitting in a dark room and someone has a torch, and if they flash it, and you go, oh, it was over there, wasn't it? But it's gone out now. And then it flashes somewhere else in the room. And that's what happens in the deep sea. These animals have to cope with these flashes from unknown distance and decide what to do.
I want to talk about some of the stuff that's come out of your lab particularly. In 2017, you discovered a new type of eye cell in deep sea fish, this rod-like cone specialised for dimly lit environments. Can you tell us about that?
I think anyone who can remember their biology back at school, you remember that the eye has these two types of photoreceptors, the rods and the cones. And in general, the rods are for dim light vision. The cones allow animals in general to have colour vision. So, we've got three cones. We've got our red, green and blue. Hence, our whole world is RGB. In the deep sea, not that long ago, people thought, all of these fish are just going to have rods, aren't they? Because, rods see in the dim light and it's dim in the deep sea, so they're just going to have rods. And that's actually true for a lot of deep sea fish. They do just have rods. But then, we've found over the years that several of them have cones. And then you have to worry about the distinction between, what is a cone and what is a rod. Cones were labeled as cones because they're sort of cone-shaped. And rods are labeled as rods because they're, you guessed it, rod-shaped. But the distinction there is breaking down. A number of animals, including things like snakes, have sort of re-evolved colour vision, but using rods, not cones, and then other animals use cones for dim light vision. So it's become a bit of a mix and match situation. What we're finding increasingly in the deep sea is it's not as simple as we thought it was.
Yeah. So what about fluorescence? Is that something you've worked on?
It is. I've got a particular thing about fluorescence because one of the things that we do as humans is we like flashy stuff and we like things that glow in the dark. So We wave UV lights around and we see things that fluoresce. You know, go to any nightclub and you've got people wearing fluorescent tattoos, all sorts of things fluoresce, you know, who actually use it in everyday lives. And then as soon as you see an animal that fluoresces, you think, oh, that must be relevant to the animal it's trying to stick out. Except you need the light to excite it to fluoresce. And in the deep sea, there isn't much. You know, in the mesopelagic, when you're in the sort of blue zone, it turns out that blue is quite a good fluorescence exciter. And yeah, in that kind of zone, it's possible that the blue light that's remaining excites fluorescent coral. And, you know, I've seen this myself diving. You go to, let's say, even just 20 meters, you're scuba diving. And all scuba divers know that when you're at 20 meters, reds disappear because water filters out red first. So if you've got a red snorkel, it kind of looks muddy green or black. And you have to come back up to the surface waters to re-see your red snorkel. But then you see, oh, hang on, that's an orange coral I've just seen. And that's a coral that is using the blue light to fluoresce. So there's a chemical process within the coral, within one of its pigments, that pulls in the blue light, it pulls in the blue photons, and then the chemical re-emits photons in the red or the orange. And it's always in that direction. It's always from shorter wavelengths, the blues and the greens, to the oranges and reds and stuff. And yeah, a number of corals, animals do this. But no, there's stuff out there that just fluoresces. I mean, your teeth fluoresce. Is that relevant? Probably not. And yeah, so that's the thing. Don't get too excited about things that glow in the dark because they may not glow in their natural habitat.
Scientific stories. Your career has been pretty damn impressive for all sorts of reasons, but the next two I'm going to pick out is the manta shrimp story.
Oh, yeah.
Right, the manta shrimp can see in 12 color channels. I think that's phenomenal, and how that sort of branched out into all sorts of different applications. I think possibly even the Blu-ray DVD.
Yeah.
Another one that sticks out is the color-changing cephalopods are actually colourblind.
Yeah.
I think those two are stories which will go down in the great things about marine science.
To use a nerdy science joke here, they are at two ends of the spectrum.
Well, hey.
One's got 12 visual channels and the cephalopods only have one. And yeah, they're pretty amazing in both respects. Mantis shrimps, they do have these, the ones that live at the surface have these 12 spectral sensitivities. As I mentioned already, we've got red, green and blue, RGB. That's perfectly adequate for seeing most colours on Earth. Birds extend that to 4, and that allows them to see deeper into the UV, the ultraviolet. And, you can show theoretically that 4 really is pretty much what you need. So stomatopods, why do they need 12? Well, they seem to encode colour differently. But then, yeah, as you say, that's the other end of the spectrum, the cephalopods that have this superb camouflage ability, completely colourblind. So how do they do it? And this includes things like giant squid, which we could argue are pretty deep sea or at least mesopelagic, and other cephalopods that live in the deep. They have this single visual pigment, but then the cephalopods that live on the surface have a single visual pigment, despite the fact that they have available to them the full spectrum of light, and despite the fact that they put on body colours and shades and texture that allows them to disappear instantly. So how do they do that? Now, I played a mean trick on cuttlefish in Plymouth once. We decided to put cuttlefish on aquarium gravel. So we bought bright yellow gravel and we bought bright blue gravel. And we took the cuttlefish out of their natural murky green habitat and put them on bright yellow gravel. First thing, they don't go bright yellow. So they're not just matching a single colour. They don't go bright blue. But what do they do? And if you actually mix bright blue and bright yellow gravel, and take a photograph with a black and white camera. Black and white camera only has a single spectral channel. You can't tell the difference between blue and yellow anymore. And this is exactly what happens to the cuttlefish. So you put them on bright yellow and blue gravel, and they put on a body shade, which is just even all over. They don't try and match the texture. So what that tells us is that they're not seeing the colour contrast that we can. They believe they're on an even grey shade. So we fool them into doing the wrong thing with their body colours. It still leaves the question of how the octopus, goodness gracious, how do they do that? And it's basically through evolution. So octopus and cuttlefish are superb at matching the intensity, the overall pattern that's provided by the intensity, and to some extent the texture, so they can raise their body skin to look like seaweed and stuff. And they do those 3 things, but they don't do colour. And all they have to do is recognise that the bottom of the ocean is usually kind of green and brown. It's not usually bright blue and bright yellow. So all they have to do is put on a sort of greeny, browny background colour, and they don't need to worry about not seeing it because they disappear.
I always thought they were actually mimicking it. They're just going for what they think it should be.
They just kind of do it because that's what evolution tells them the ocean floor looks like.
So are deep sea animals doing anything different in the brains? Are their brains evolved in any particular way to process this type of information? So, what's going on in the brain department?
Oh, good question. And the brains are not that different. They're, again, similar to those of surface living fish. They don't have the bits that need to process colour, unless you're a weird deep sea fish that could process colour. Cut back to brains, they're not that dissimilar. They can become simplified for certain areas that don't need as much light as the surface dwellers. But they have all the different, the metallencephalon, diencephalon, to use the fancy terms. The eyes go to the same sort of area of the brain to allow the brain to interpret images. But I'm kind of arm-waving, as you can tell. Really, the real answer is, although there's overall similarities, we just don't know.
You once told me that you lived underwater for quite some time in the Aquarius laboratory.
This thing doesn't exist anymore, but the Aquarius habitat was basically an underwater caravan on the Florida Keys. And, lucky marine biologists could go and spend 10 days underwater. It doesn't mean you have to be in the water for 10 days. You can duck up into this caravan, which was held at about 25 meters. So you can duck up into dry-ish air and dry off-ish. Now, humans are not adapted to being that damp that often. And you do start growing fungus. It's not pleasant.
Didn't you just describe it once as like having all-over-body trench foot?
Essentially, yeah.
Athletes, everything.
Yeah, and when I came up, I got what's called a skin bend, which meant that a lot of the skin of my hands and feet just fell off all in one go. So I had this sort of foot pad, which just peeled off, which was very unpleasant. But I dried it out and kept it in my wallet for a bit, but I gave it to a girlfriend. She was very happy with my foot parchment, that sort of thing. It was fantastic. I definitely felt better when I was out in the water. And, the good thing about the habitat was that you could spend 8 hours a day out in the water and you just start thinking like a fish. But it was a great experience, great privilege to live amongst the fishies and the other animals for that amount of time. So, yeah, it was fantastic, apart from the smell.
That's been fascinating, Justin. Thank you very much for your time and thanks for coming on the podcast.
Yeah, my pleasure, Alan and Tom. It's been great to talk to you about things that I love.
Hi, I'm Claudia. I'm a university student from Sydney, and I wanted to thank you for your wonderful podcast and ask you a question about snailfish. How did they get their name? I know they have a sucker on the underside of their bodies, but what is it used for? And is it the only reason for their name? Thank you.
They're a super successful family. Snailfish, the Leparidae, are found from like the intertidal, you can find them in rock pools, to being the sort of deepest living fish, at least that we currently know about. They have adapted their pelvic fins into a little sucking disc that lets them hold on to smooth rocks and to the fronds of kelp and seaweed. And when they're holding on there, they tend to curl their little body around and they look like a little snail. And that is where the snailfish name comes from.
Hello, this is explorer oceanographer Don Walsh. And the subject for today's program is windows in the sea. When using manned or unmanned submersibles, you need to be able to see outside. But these windows, or viewports, are also pressure boundaries, where the observer inside is at atmospheric pressure, and outside it's at higher pressures that vary with depth in the sea. It's an engineering challenge. The viewport material needs to be strong, so the trick is to have something that's also transparent. For over 2 centuries, glass was the logical choice. In fact, the only choice. From an engineering point of view, glass is very strong, but there are serious problems with it. It is unpredictable. This is mainly due to the fact that it loses strength when it is notched and then can fail without warning. I can give you an example, and that is cutting a piece of glass for a picture frame. You make a scratch across the piece of glass, and then you just tap the glass very slightly, and it breaks along that scratch line. And that's called a stress concentrator. Well, it's not very good for a window in the sea. If somebody happens to scratch it in some way, now you've got a stress concentrator, and it could fail without warning. But it was all we had until the 1920s. And that was when another stronger material was developed. It was called fused quartz, a transparent material made from very pure silica. By the late 1920s, General Electric was able to produce larger quartz glass viewports. They were difficult to fabricate, but were much, much stronger than conventional glass. But fused quartz had some of the same limitations, namely the problem of stress concentrations. And again, you could get sudden failures. In short, it was tricky. But at Bermuda in the early 1930s, Dr. William Beebe planned deep diving operations with his bathysphere. For his windows, he used 3-inch thick fused quartz disc. Nevertheless, with these windows, he bravely got to 3,028 feet in 1935. Dr. Beebe was indeed the first hydronaut. The last use of quartz glass in a submersible was by Beebe's partner, Otis Barton. In 1949, he dove his new design to 4,500 feet. However, by then, fused quartz had been eclipsed by a newer and safer material called plexiglass. It was invented in 1933 in Germany. It's not a glass. It's a plastic material. That means that it can flow under pressure, not like water, but very slowly adjust to pressure changes, and in that way avoid any stress concentrations. And it can also resist surficial damage. You could actually take a knife or sandpaper and scratch it without any loss of strength. In the late 1930s, plexiglass really made it possible for inventor Auguste Piccard to begin design and construction of bathyscaphe manned submersibles, which had unlimited depth capabilities. In 1960, the two 7-inch thick acrylic viewports on the bathyscaphe Trieste worked perfectly on a dive to a depth of nearly 7 miles. There, the outside pressure was an astounding 8 tons per square inch. By the 1970s, hundreds of manned and unmanned submersibles had been put into service worldwide. All used plastic viewports. While acrylic viewports were the beginning, it did not take long for entire submersible hulls to be constructed of massive acrylics, where the hull is essentially one big window. We humorously call this experience being inside a people bowl. That is, you're in the bowl and the fish are looking at you from the outside. So until next time, think deep, and thanks for listening.
And that concludes this pressurized version of the Deep Sea Podcast. If you've enjoyed this episode and you'd like to go into some more detail, you can find the full episode in the feed. Just match the episode numbers. We'll deep sea you next time, and I abyss you already. The Deep Sea Podcast is supported by our company, Mantis Oceanic. If you'd like to explore the deep sea for yourself, we can provide the technology and know-how to allow you to do that. Or if you'd like to bring the deep sea to your audience through storytelling, fact-checking, or presentations, we can help with that as well. We want the deep sea to be accessible to everyone.